Hi, I'm Brianna, and this is Decoding Physiology, a series from Decoding DX where we focus on the pathophysiology behind various clinical entities and conditions to help you better understand, because we believe that if you understand the why, you'll make better decisions for your patients and learn for the long term. This is part 4 of a multi-part series on metabolic alkalosis. These videos are building on each other, so if you haven't watched parts 1 through 3, please go back and watch those first. All right, so we already talked about if you have a reduced GFR. So what if it's not reduced? Or what if you address that issue and you still have an alkalosis going on? Diuretics can affect this, so we're going to start with a simpler effect first where you don't have a diuretic on board. The next branch point is looking at the volume status, hyper or hypovolemia. This can be based on objective findings, subjective findings, history, or the urine chloride. And you'll see why that urine chloride can be a really helpful indicator when we get into the physiology later. We're going to start by looking at a volume up state or a hypervolemic state. Some of the main clinical entities that will lead to this are any sort of hyperaldosteronism. This can be primary hyperaldosteronism, this can be high levels of renin, anything that's leading to a hypervolemic state in the presence of aldosterone. Okay, so this is where the physiology really dives into renal phys, so buckle your seatbelts and we're going to have some fun. So to start off with, we're going to go through the normal physiology. In the distal convoluted tubule and the collecting duct, we have two main broad categories of cells. In the principal cell, we have a sodium-potassium pump that creates a gradient that pulls in sodium through the ENaC channel into the principal cell. This then creates a negative lumen charge, which attracts the potassium into the lumen of the nephron. Let's go through that animation again real quick. We have the sodium-potassium pump powered by ATP that creates a gradient inside the principal cell. This gradient pulls sodium in through the ENaC channel, which creates a negative charge in the lumen. This negative charge, in addition to a concentration gradient, pushes the potassium from the cell into the lumen. Meanwhile, in the intercalated cells, there's a reaction that we're familiar with, yay! There's carbonic anhydrase catalyzing the reaction of water and carbon dioxide to create hydrogen ion and bicarbonate. These two are exchanged in a way that we've seen before, where the bicarbonate is pumped into the blood and the hydrogen is pumped by an ATPase into the lumen of the nephron. We can go through that one again as well. In the intercalated cell, we have the reaction we're familiar with catalyzed by carbonic anhydrase. The bicarbonate is going to be pumped into the blood in exchange for chloride, and the hydrogen ion is going to be pumped into the lumen by an ATP pump using ATP. In a normal physiologic state, when we have low flow to the kidney from the point of view of the kidney, this is going to trigger the renin-angiotensin-aldosterone system, so we will have the presence of aldosterone. Aldosterone is going to enhance all of the different actions that we just talked about in the principal and the intercalated cell. Sodium reabsorption is tied to potassium excretion in the principal cell like we saw before. Aldosterone is also directly related to hydrogen excretion in the intercalated cell and the production of new bicarbonate that is sent into the blood. But in a relatively low flow state, there's overall less sodium and water delivered to the distal convoluted tubule and the collecting duct in the first place, which effectively blunts the effect of wasting potassium and hydrogen and the related process of bicarbonate production. When volume is restored, we go back to the normal state. To summarize, if our kidneys are sensing a low volume state, they're going to tell the body to release aldosterone, because aldosterone is going to trigger the activities in the kidney that are going to absorb sodium and therefore water. The absorption of sodium in the distal convoluted tubule and collecting duct is directly related to the excretion of potassium, the excretion of acid, and the production of new bicarbonate. This is not reabsorption of existing bicarbonate. This is production of new de novo bicarbonate. Let's say we have the opposite situation, again with normal physiology. If we have high flow to the kidney, we're going to have aldosterone turned off. So we're not going to have an excess aldosterone state. In this high flow state, we're going to have high levels of sodium and water delivered to the nephron. But because we have aldosterone absent, all of the processes that we've talked about are going to occur at a much slower rate. So these processes are still happening, but they're happening much slower. Overall, there's going to be less sodium and water reabsorbed, And this is going to slow the entire process down because everything depends on that amount of sodium reabsorption in the principal cell. So we're going to have more sodium excreted, less potassium and hydrogen excreted, and less de novo bicarbonate produced. Okay, so let's say we're in an inappropriate aldosterone state. This is the pathophysiology that is on that part of the diagram, the big intimidating diagram that we saw earlier. An inappropriate aldosterone state means that we have aldosterone active despite there being a high volume state. Normally, with normal aldosterone, it's triggered in a low volume state. The pathophysiology of inappropriate aldosterone combines the two previous scenarios that we talked about with the low flow and high flow states. So we have aldosterone present, and the aldosterone is going to increase all of the actions that we've discussed so far in the principal cell and intercalated cells. But in this case, we're going to have a high delivery of sodium to the distal nephron, which is going to feed the accentuated processes turned on by the aldosterone. So we're going to have an extra amount of sodium reabsorption, which, as we talked about earlier, is directly tied to the other processes. So we'll have an even extra negative charge in the lumen, which is going to strongly attract extra potassium, leading to more potassium wasting. Aldosterone also directly accentuates the process in the intercalated cell, where we produce de novo hydrogen and bicarbonate. So these processes are going to occur at a higher rate as well, leading to excess excretion of hydrogen and excess production and absorption of new bicarbonate. So in the end, we have extra sodium reabsorption, extra potassium wasting, extra hydrogen wasting, and extra reabsorption of free bicarbonate, leading to a metabolic alkalosis. Let's walk through those animations one more time. So we have a high flow state with the presence of aldosterone. Aldosterone is going to accentuate the activities that we talked about before with all of the different ion pumps and channels. But we're going to have a high delivery of sodium and water to the distal nephron in this state. So we're going to have accentuated amounts of all of the actions: extra sodium reabsorption, extra negative charge in the lumen, extra secretion of potassium because of that negative charge, extra production of hydrogen ion and bicarbonate, which are then pumped into the lumen and into the blood, respectively. So in the end, we have extra sodium reabsorption, extra potassium wasting, extra hydrogen wasting, and extra free bicarbonate that is made new that is absorbed into the blood. But we also have to maintain the alkalosis. There are two main processes in this state that will contribute to maintaining. To start off, we can just have a continuous process. For example, if someone has an aldosterone secreting tumor, primary hyperaldosteronism, or hyperrenism, you can just have a constant state of high volume in the presence of aldosterone, leading to the process we just went through happening without stopping. That will lead to excess bicarbonate being produced and not giving the body a chance to compensate. We're also going to have a hypokalemia because one of the things that we talked about with the principal cell was that the more sodium that's reabsorbed from the aldosterone, the more negative charge is in the lumen, which then attracts extra potassium to be wasted in the urine. So we'll have a hypokalemic state, which will trigger the compensatory actions that we talked about in part 1, which means that we have a shift of hydrogen from the blood into the cells, leading to a relative decrease in the concentration of hydrogen, which contributes to the alkalosis. Here are main references, same as last time. This one was pretty complicated, so I encourage you to go back and watch through and make sure that you understand, because the next few videos will build on this one. Join us in part 5 as we move on to the next pathophysiologic mechanism of metabolic alkalosis.